My name is Rashna Sharmin. I am a PhD student in the biology department. I want to talk about program cell death. In multicellular organisms, cells that are no longer needed are destroyed by highly regulated cell suicide mechanism called program cell death. Some of our cells are really complex in that they have different compartments, such as neuron. Intracellular trafficking is one of the unique feature of complex cells such as neuron, as different organelle needs to be transported to distant part of the cell. And one of the important organelle is mitochondria, and mitochondrial trafficking is very important in complex cells such as neuron. So our central question is, how does intracellular transport of mitochondria affect of the death of specialized cell? We use C. elegans as a model system for our study. One of the many great advantages of C. elegans is it has transparent body, which allows us to see the cell dying in a living animal. We use cells like epithelial cell, which dies embryonically in C. elegans as a model system to understand how complex cells die. And as the cells start to die, it shows some unique feature, and we call it compartmentalized cell elimination, or CCE. Through our forward genetic screen, we found that UNC16 mutant has CCE defect, and the defect is the cell persists in the larvae. So UNC16 is a C. elegans homologue of mammalian kinesin 1, which is a microtubule plasmin-directed motor protein, carries various cargo. So we asked whether mitochondria are cargo for UNC16. And indeed, we looked at mitochondrial reporter. We see mitochondria is present in the UNC16 mutant remnant. Mitochondria specific UNC16 is able to rescue UNC16 mutant CC defect significantly. We looked at mitochondrial distribution in wild type animal, and interestingly, we see as the cell develops and mitochondria moves from the process towards soma in retrograde fashion. And before the cell die, we never see mitochondria in the process. It appears to us mitochondria moves from the process to soma as a preparatory step for CCE. So mitochondria is moving from process towards soma in retrograde fashion. We ask why mitochondria is not able to come back to the process. So we looked at cetricaspase mutant, and cetricaspase is a cystin protease that is a crucial component for programmed cell death. And very interestingly, while in cetri not full loss of function mutant, we see mitochondria in the process of the tail spike cell. We suggest cetricaspase may be targeting a motor protein that is able to carry mitochondria from the soma towards process. To find out that motor protein, we performed a candidate gene screen, and interestingly, we found UNC104 kinesin 3, which is another macrotubule plasmin-directed motor protein, maybe a relevant cetricaspase substrate. And one of the 10 caspase target sites of UNC104 shows CCE defect. Since UNC104 is not traditionally known to carry mitochondria, we validated mitochondria as a non-conventional cargo for UNC104 kinesin 3. Mitochondria specific UNC104 was able to rescue UNC104 CC defect. We see localization of mitochondria UNC104 in the tail spike cell. Also, we found UNC104 is involved in anterograde mitochondrial trafficking, while in citric caspase and UNC16 mutant, we see a lot of mitochondria in the process, while with mutated UNC104, there we see less mitochondria. We suggest in presence of UNC104, a lot of mitochondria is coming from the soma towards process, but in absence of UNC104, it's not able to come in the process that much. Since we are saying UNC104 and UNC16 both are involved taking mitochondria in opposite direction, and both are microtubule plasmin-directed motor protein. For our model to be true, microtubule polarity has to be mixed microtubule polarity in the tail spike cell. And from this chymograph, you see that we found tail spike cell has mixed microtubule polarity. With white arrowhead, we see retrograde microtubule, and with the yellow arrowhead, we see anterograde microtubule. This is our overall model.